Hello, everybody. My name is Caitlin, as Helen said. I work for Cure Parkinson, so I'm our research communications officer. And I'm basically going to be hopefully helping to break down some of the science that we'll be discussing today, because the endolysosomal system is a big, big word that even I didn't really get when they threw that at us. So I'm going to try to break it down. This will probably be a bit of review for many of you, as I know many of you are very well versed in the science. But hopefully it'll provide some insight and some new little things. So I will start with actually just what is Parkinson's. And so Parkinson's is a neurodegenerative condition that is characterized by the progressive loss of dopamine-producing neurons in the part of the brain controlling movement. And that is a big, long definition with a lot of very boring words. So I'm going to hopefully put it in a bit of a better way. So we know that our brains are made up of lots of different types of cells, and one of the most common are something called neurons. And so neurons communicate using these special signaling molecules called neurotransmitters. And so one of the most infamous is dopamine, which we'll discuss on the next slide. But basically, they use these to talk to one another, and this is how sort of actions and things take place. And so you can kind of think of it like calling somebody. So if you ring somebody, but they don't have a signal, they're not going to receive the call. And in a similar way, if a neuron wants to communicate with another neuron and there's no neurotransmitters, they can't complete that call. And so that's sort of what you see in Parkinson's as we lose those dopamine producing neurons, is you don't have enough dopamine for them to communicate with one another and carry out that intended function. So dopamine is one type of neurotransmitter. It's pretty popular in pop culture, mostly due to its involvement in the kind of reward and motivation system. So people with ADHD have a lack of dopamine in a specific part of the brain. But in a similar way, dopamine also plays a very important role in our movement. And so it sits at the very beginning of a sort of movement cascade in a part of the brain called the substantia nigra. And so this part of the brain has just a ton of dopamine producing neurons, and it makes a bunch of dopamine that is then used by other neurons in other parts of the brain and further down that pathway. So it's kind of like a bunch of dominoes. You set one at the beginning, which would be the substantia nigra, and it sets off that chain of events that lets me sit up here and talk to you all and move my arms and everything. And so that's sort of how that works and how dopamine works. But now we're gonna jump into the cell. So I'm gonna need you all to step back with me into like 7th grade biology. And we're gonna go over the parts of the cell. I'm gonna move the little thing up here out of the way. And so cells, including neurons, are all made up of a bunch of parts. We call them organelles, which is a fun little name. It's just organs, but smaller. And so they all work together with one another to keep it functioning. The most common and the thing that stuck with me, which I found out that British people don't learn this analogy when I was in the UK, is that the parts of the cell work like a factory. And so each individual part has a very specific function that helps it basically do what it needs to do. And so just kind of an example of some of these, many of which you've probably already heard of. So the nucleus is sort of the CEO of the cell, so it's the Helen of the cell. And it has all of our genetic information stored in it, so our DNA. And so from there, it basically controls what the cell does. It tells the cell what to do, and it does that by reading that DNA. We're going to go into genetics in a little bit. But another part is proteins. And so these are sort of like the workers of the cell. They're little molecules that carry out most cell activities. So these include things, you'll hear them referred to as hormones, enzymes, antibodies. These are all types of proteins that all have very specific functions. And so they sort of do a lot of things. They're built in a specific part of the cell, and our DNA often contains the instructions for building those proteins. And then the last one, which we'll jump into a little bit further, is something called the lysosome. So these are the garbage men of the cell, very fun job. And they handle the breakdown of cellular waste. And so the way that they do this Well, here we go. In Parkinson's, many of these cell pathways don't function the way that they should. And so it's not limited to just the lysosome. It can often be many different parts of the cell that are not going well. And so if a neuron can't do what it needs to do, then unfortunately it will die. And so that is sort of what we see in Parkinson's, thus the loss of neurons. So the pathway that we're going to sort of zero in on this week is the waste disposal system, so things involving the lysosome. So what actually is cellular waste? Because that's just a word. It doesn't really mean much. They don't have trash like we do. But they need to dispose of dysfunctional, damaged, and or inefficient parts of the cell in order to keep it healthy and working. So similar to us, if our room is full of trash and clutter, and we can't even walk from one side to the other, we can't really do what we need to do. So in the same way, the cell needs to sort of get rid of those things and keep everything sorted. But how exactly is that waste created? So one way it could be created is, gotta move this one again. There we go. One way it's created is things like proteins could be built incorrectly. So I've made an analogy to hopefully something that Americans, if you've seen the show Drake and Josh from Nickelodeon, there's a very infamous episode where the two brothers build a treehouse and they realize in the treehouse that they did not bring a buzzsaw in and they did not make the door. So they actually can't leave the treehouse. So it is built incorrectly, and the rest of the episode is them attempting to get out of the treehouse because they also don't have phones. And so, similar to that, things like proteins might not build correctly, meaning they can't function as they should. So it's not gonna be a very good treehouse. Another thing is cell organelles just become less efficient over time. And so similar to a lot of things and just cells in general, as they age, they're not going to work as well. And so things need to be replaced regularly in order to keep up with that. And then another way, it can also be a byproduct of reactions. So we know that there's a lot of little reactions in the cell to make various different things, including cellular energy, which is what that diagram is right there. And so when they make these reactions, oftentimes there will be sort of waste that is created as just a byproduct. So things react, but maybe not all of the molecules are used up. So instead, they're sent away. And so we'll go into a little bit more detail about what one of those byproducts might be in a second. So why is getting rid of that waste important? Well, resources in the cell are limited. And so there's not an endless supply of everything for the cell to use. And so they need to use it basically as efficiently as possible in order to keep everything sorted. There's also some types of waste that could be more dangerous than others. So, you can think of it kind of like nuclear waste versus like just a garbage can. One I would argue is probably a little bit more dangerous than the other. So one of those is something called reactive oxygen species. So these aren't an actual animal or anything. They're just these little molecules that are quite reactive. They're produced as a byproduct of energy production in the cell. And oftentimes the cell will just sort of get rid of them. But when you have lots of dysfunctional energy production, you'll get quite an excess of them. And if the waste disposal system in the cell isn't working either, then it can't get rid of it. And you can lead to something called oxidative stress, which is actually combated by something called antioxidants. So you often see on like vitamins and things, it's like, it's a good antioxidant. I didn't know what that meant. Now I do. So basically that means it can reduce oxidative stress. Another thing is alpha-synuclein. So alpha-synuclein is a protein that is commonly misfolded. So proteins are made up of these long chains of molecules called amino acids, and similar to like origami, you have to fold it a very specific way for it to do its function. And so oftentimes in Parkinson's, we see when the cell tries to build and make alpha-synuclein, it doesn't quite do it correctly and it gets a bit misfolded. And so basically what it tries to do then, it builds up and they're quite sticky. So they stick and clump together to form things called Lewy bodies, which impede the cell from being able to do things. And so that's another type of waste that might be a little bit more quote unquote dangerous than others. And then we know that some parts of the cell, such as the mitochondria, can initiate cell death pathways when damaged. And so when things like reactive oxygen species or alpha-synuclein build up, can actually lead to damage of other parts of the cell. So the mitochondria is the powerhouse of the cell. It is what creates all of the energy. And so when it gets damaged, it can release these little molecules that sort of trigger a cell death pathway. And in the cell, when that death pathway starts, it very rarely can be overturned. So it's just it goes on and you can't stop it from happening. So we want to prevent it from, not killing itself, basically. So you want to prevent it by that. So how do we actually get rid of waste then? Well, that's where the lovely little lysosome comes back into play. So these are the garbage men of the cell. They taxi around little enzymes to break down cellular waste. So enzymes are a type of protein. There's a special type of enzyme called hydrolases, which are the ones in the lysosome. But you can think of enzymes sort of like an escalator versus stairs. So they're proteins that can speed up reactions in cells. And so, you could still have the reaction, such as you could still take the stairs, or you could use an enzyme / an escalator and get there a whole lot faster. And so it sort of helps with that. So it speeds up the breakdown of that waste so it doesn't accumulate. So there can be a number of reasons why the waste removal system isn't functioning properly. This can include issues with the lysosome itself. So is something blocking it from going where it needs to go in the cell? Enzyme trafficking. So the enzymes themselves aren't built in the lysosome. They have to be transported from one place to the other. So something blocking them from being transported into the lysosome. And then just issues with the enzymes themselves. So as I already said, sometimes there can be issues when we build proteins that can lead to waste and such. And so are they being built correctly? Is there enough of them to carry out the task that they need to do? And this sort of leads into the next little part, which is the building of enzymes can often be influenced by genetic factors. Ooh. All right. So genetic risk factors for Parkinson's. So genes are sections of our DNA that direct cell activity, most notably by providing the instructions for building proteins. And so basically the cell will read these little sections of the DNA, it'll export it into the cell, and then another part of the cell will read that instructions and build the protein as such. So changes, also referred to as variations or mutations in genes, can lead to changes in the protein it is intended to make. And this can lead to that protein functioning less efficiently or incorrectly, depending on how bad the variation is. And so there's a couple different ways they can do that. You can add an extra thing into the code. You can take something out of the code, or you can replace one with another in the code. And so variations can be inherited, but they can also occur randomly. So you'll often hear sort of familial Parkinson's is usually associated with some sort of genetic risk factor. It is important to note that they're just risk factors, so none of them currently will determine whether or not someone will go on to develop Parkinson's, but more so a contributing factor, so it'll raise the risk. So you can kind of think of it like messing up a recipe for cookies. So messing with the genetic code, you'll still get potentially a cookie in the end, but it might not be exactly what you want. So for instance, if you forgot the eggs in it, that could be sort of like taking out a part of the code, so you've missed a step, and you're not going to get what you want. Too much flour would be like adding something into the code, and so you have too much of one thing and it doesn't quite end up the way that it should. And then too little butter, for instance, would be like taking something out of the code. Or baking powder instead of baking soda, which would be replacing one of the parts of the code. So that's a good way that I think of it, at least. And so we know there are several genetic variations that are associated with a higher risk of developing Parkinson's. I've pulled out 3 below, but there are much more than this. And so many of these variations affect proteins that are associated with that waste disposal system that we mentioned. So one of them is GBA1, and this codes for the lysosomal enzyme GCase. I don't remember off the top of my head the full name of that enzyme, but you don't even know that. So that helps with breaking down waste. Another one, which a lot of the presentations today will go over, is something called LRRK2. And so this codes for the protein kinase, LRRK2. And so oftentimes we'll see that being overactive when that mutation happens. And then the last one is PINK1 and Parkin. So these are two proteins that are involved in the mitochondria recycling pathway. So getting rid of old mitochondria and replacing them with new ones. And so the ones that we're gonna hone in on today are LRRK2. I believe we also have a presentation on GBA1 as well. But that's sort of, I guess, our scientific introduction into it. I think I managed to finish a little bit early, so I will open it for questions, but if not, then we'll just do a little movement break until our next one. Yes.
I'm real impressed with the cookie slide. I wonder if you bake those cookies yourself or.
Oh, no. I will say the question was, did I make the cookies on the cookie slide? I didn't make them, though I will say I am notoriously a bad cook, and there is a very good chance that I have made any one of those at some point in my career.
My question really is, and I don't know, what is the function and role of that asynuclein protein? You know, we hear a lot about its dysfunction. It's folding wrong, it's getting sticky, and it's interrupted. But when it's working properly, do you know what its function is?
So the question is, what does alpha-synuclein actually do when it's working? And they're not really sure at this time. So they have some sort of hypotheses, but since it's rather hard to study neurons and such, they're not sort of conclusive. Some have suggested that it may have a role in the immune system and sort of triggering an immune response within the cell. And so that's kind of one suggestion for it. We do know that it's found in higher concentrations in neurons than maybe other cells, but I don't believe there's anything conclusive at the moment about it. So that just makes it even better. Any other questions about this or any sort of just general Parkinson's?
It's worth saying you do need a bit of alpha-synuclein. You just don't need as much as seems to be happening.
Yeah.
Because that's when it starts to clump and it causes further toxicity.
Yes.
What is a kinase?
Kinase, Alex might be better suited to answer this one than me, but it's just another type of enzyme. What specifically it does.
So just off the top of my head, a kinase is essentially a type of enzyme that helps other enzymes work properly. So it's kind of like this perpetuating machine. You need one enzyme to do whatever it needs to do, but you also need enzymes to help other enzymes. And then you need another enzyme to help that enzyme. It's just, you need a lot of enzymes within the cell. I think the running count is around 100 or 200 enzymes that are constantly present in a cell helping other enzymes do their function.